Phatnani, and it's my pleasure to welcome you today to the evening session on ALS, Genetics and Genomics. I direct the Center for Genomics of Neurodegenerative Disease here at the New York Genome Center, and I'll give you a brief overview of what it is that we're trying to achieve. So the CGND, because Center for Genomics of Neurodegenerative Disease is quite a mouthful, has three main goals: to serve as a hub for collaborative interactions, to build tools and resources for the community, and to apply genomics to understand the molecular basis of disease. And the way we do this is by integrating studies across multiple scales, population, tissue, cell, etc. You'll see in a second what that means. We also integrate multiple data types: clinical data, whole genome sequencing data, phenotyping data, histopathology data, any kind of data that we can intelligently mine to link up together so that we can better understand how disease sets in and how disease-associated mutations affect molecular mechanisms that go awry. In pictures, this is what that looks like. So there's two halves to what we do. For the genomics, we study patients with their clinical information. We collect DNA to generate whole genome sequence information. We use their post-mortem tissues to look at how these genes are expressed in the central nervous system. We also take cells derived from these patients that we can study more closely in a dish. In parallel, we also work with mouse models of the disease, study cells by themselves in a dish, or look at tissue sections. And what do we do with the tissues and the cells? We look for how gene expression is altered in space and time so that we can identify networks of molecular pathways that are going wrong in the disease. And this is why integrating across scales is important, because studying them in a dish gives you detailed information about what can go wrong. Relaying that information into what's going on in tissue tells you what does go wrong. Linking the information from mouse models to what's actually happening in patient tissue tells you what's relevant from the mouse studies to the human system, and then syncing that back up with the genetics and the clinical characterization is really important because ALS is a complicated disease. The challenges with ALS genomics are essentially twofold. It's clinically heterogeneous, which means it can present itself differently in different patients, can set in at different places, it can spread with different patterns and different rates. The other thing is that to get large numbers of samples that are to do statistically well-powered studies, because ALS is relatively rare, you need to pool together samples from multiple different clinics. Even the busiest clinic in New York is going to see a few hundred patients a year, and you need few thousands to be able to integrate studies at this scale. And in order to do that, you need to be able to work together. None of the stuff that I'm telling you about today would have been possible without Lyle and Matt and Neil, who are going to tell you more about what each of them do. I work very closely with Matt on this aspect, very closely with Lyle on this aspect, and my background is in molecular biology and biochemistry, which allows me to do this aspect. So I told you that we serve as a hub for collaborative interactions, and that's one of the ways in which we envisioned a solution to overcoming the challenges of the sample size issue. We built a federated program with centralized governance, and by that I mean we are a centralized hub to coordinate administrative processes, sample processing workflows, clinical data collection, how and when data is collected at various sites, how and when data is transferred to us, how we disseminate the data. And in order to do all that, we also need to work out data warehousing solutions. So because we have this centralized coordination that works in close partnership with other nationwide ALS efforts, we can make sure that multiple participating sites are all collecting samples and data in a way that can be studied together. Because different clinics are going to measure different things in different ways at different times. And this kind of coordination is critical if you want to ultimately be able to pool things together. So to give you an idea of the kinds of ALS partnerships we have at NYGC, here are all the sites in the United States from whom we receive samples and data. These partnerships extend worldwide. There are currently 30 partners in 5 different countries. And another key aspect of this is for the data to be as useful as possible to as many people as possible, as quickly as possible. We also make sure that the data are shared as quickly as we can generate and QC them. This brings people who have new analytical tools closer to the data much quicker. They don't have to wait for it to be published. And this in turn accelerates the science. So these kinds of partnerships have led to the identification of several additional ALS-associated genes. And studies of the tissue samples have led to better understanding of how ALS-associated mutations are affecting gene expression and what that might mean for disease-associated pathological processes. I'm not going to go into detail about the science because I want to leave the floor for Neil, Matt, and Lyle to tell you about what it is that they do, but I'd be happy to answer questions about the science later. I want to say thank you to the patients who participated in these efforts, to all of the organizations whose support and encouragement were critical in getting these efforts off the ground, the team at the New York Genome Center and our partnering institutions who made the work possible. And I wanted to thank you for coming here and allowing us a chance to tell you about what it is that we do. I'm going to turn it over to Neil, and we'll take questions for everything at the end.
This is the 5th anniversary of the Ice Bucket Challenge. This was a transformative experience for the ALS space where over 17 million people donated money to ALS, poured ice water over their heads, and really changed the ALS Association, I think, among a number of other charities. And we've been able to, I think, much more rapidly accelerate our work for ALS as well as the quality of services that we provide for people with ALS around the country. And so we're taking this year as an opportunity to reflect on what happened and what we've done since then and how we can move forward. And so we've asked Hemali to speak about that on a video, which is part of our 5th anniversary campaign. And so I want to share that with you now because she was kind enough to do it, and I think it turned out really well.
For the people who've participated in the Ice Bucket Challenge and donated money, what you've made it possible for us to do is transformative in the field of ALS genomics. We're working together in a way that we haven't before. Everybody's on board. We're sharing in a way that wasn't conceivable 5-7 years ago. Gifts from the Tau Foundation and the ALS Association that was enabled by the Ice Bucket Challenge essentially allowed us to establish the Center for Genomics of Neurodegenerative Disease. We use genomics to try and understand the causes and mechanisms underlying diseases such as ALS. Without the Ice Bucket Challenge, none of this would have been possible. There are many challenges to finding treatments and a cure for ALS. We need lots of people working together, and we need these tools and these resources to be available quickly and broadly. If you're going to rely on a single clinic to collect the thousands of samples that you need, it's going to take you several years. So you need to be able to pool samples from multiple different sites. We've built one of the largest resources of ALS whole genome sequencing data. This resource has been shared with partners all over the world. It has accelerated the pace of ALS gene discovery. It's led to the largest ALS sequencing study in the United States. We also share data with people in Europe, and we get samples from sites in Europe. So this is not just a nationwide effort, this is a truly global effort that empowers researchers all over the world, all because of Ice Bucket money. Before such broad data sharing, all of this would have taken much, much longer to do. We're linking these changes to disease-related processes at a depth and scale and in detail that hasn't been done before. What this means is that each dollar goes further because you can do so much more with one sample. What we've done is a fantastic beginning. I challenge you to help in whatever way you can. Join a walk team, volunteer for events. Let's share with even more people around the world the data that we've collected. Let's enable even more clinics worldwide to be able to participate in studies like this. Let's do it together.
Thank you. That was really wonderful. So I wanted to take a few minutes to talk about what the ALS Association is and what we do here. And our mission is to discover treatments and a cure for ALS and to empower people affected by ALS to live their life for the fullest. So we're working for people who have ALS, their caregivers, their family members, and we're working to discover cures and treatments and provide services and to empower and fight for people who have ALS. And we do this in a comprehensive way. We have an advocacy program, a care services program. We have both a local presence and a national presence as well. We support over 88 clinics around the country that provide multidisciplinary care. 66 of them also provide access to clinical trials. They provide research. We have a full research program that I'll be talking about today. And we also have a robust advocacy program. We are having a meeting in a couple of weeks where we'll be sending people to the Hill to ask Congress for better benefits for people with ALS, to expand research funding for ALS, and also we always have to be vigilant to make sure that the rights of people with ALS don't get taken away, that you maintain your benefits, that there aren't pre-existing condition clauses that crop up into our insurance practices, for example. So when we talk about research, we focus on funding the best science wherever we can. That approach makes sure that we are not invested too heavily in overhead. It lets us be flexible, but it also means that we get to fund infrastructure programs like this, like the Genome Center, that enables lots and lots of different science in different areas. We also support clinical trial consortia and other kinds of programs that help people come together and work together more efficiently. And our portfolio can be broken up into 4 main clusters of science: living with ALS or managing ALS, basic research on what ALS is and how it works and how it ends up harming people, research to identify targets for drugs and helping get those targets ready for studies in humans, and then actually funding some of those human studies themselves. And so we tend to fund $16 to $18 million a year, and some of our projects are multi-year money. So this is an example of our committed dollars, which is about now, I think, $35 million a year. And so we've been doing this for, of course, a long time, but the Ice Bucket Challenge, as Hemali mentioned in the video, has been a transformative event. It allowed us to more than triple our research spend. And we spent part of that money with an emphasis on collaboration by funding infrastructure programs like this Genome Center, and we can start to see the impacts of that. And so this is the numbers of people that we funded, these little dots here. And then the lines between them are the papers that they were writing between themselves. These are co-authorships. And the thicker the line, the more times that those folks were writing together. And then you could see in 2018, and this is partial data, you get more dots, more people that we're funding, and they're also working much more closely together. A lot more co-authorships, a lot of partnerships. And for those of you that aren't in science, These collaborations take a lot of effort, a lot of energy. I'm trying to plan an 80th birthday party for my dad with my sister. That's a collaboration. It's taking a lot of time and energy to work with my sister because she doesn't live near me. And you can imagine doing this with multiple people across multiple time zones. And that's what the scientists here are doing all the time and doing quite effectively. They've also been leveraging the funding that we've been giving them in lots of different ways. And part of our strategy is to give relatively small and short awards so that they can be used as a basis for bigger applications from, for example, NIH. And so we asked Research Triangle Institute, a think tank, to come and do an evaluation of what's been happening with our ice bucket spending. They put out a survey of the scientists. Some of you filled out that survey. We had just about 100 people respond to this question. They got $40 million of our funding over this time period, this entire time period, and they said that that $40 million they were able to directly turn into $124 million in additional funding. So we give a grant, they used that grant, did their work, and then that was the basis for getting their very next grant. And so we had more than a 3-to-1 leverage. And again, that's partial data because they're still leveraging the data they're getting from their current work. So we've been doing that. We've also, through our advocacy program, been working with NIH and the FDA. The FDA has become, I think, even more responsive to the needs of ALS and other rare diseases. And it's probably easier to get an ALS drug approved than it ever has been. And so we're making sure the science and the regulations can work together more effectively. And a lot of the folks here have been helping on that too. That's a scientific regulation process. And a lot of scientists have been working hard to help the FDA figure out how to be more efficient. So going forward, we're working on a few things in terms of the strategy for our science. So we need to find out ways to be even more transparent. And so one of those things is documenting, both for individual scientists and as funders, documenting the successes that are coming out of programs like this. And I have this note here for tenure and promotion. Hemali and I were talking about that. It's very important that people who spend their career sharing and collaborating and doing all of that hard work get recognized by their academic leadership and get promoted and go into leadership positions themselves. And so that's something we need to do. And there's a measurement element to it that we're working on. And I know the Genome Center is working on that as well. We also have to be efficient. We have to think about spending our money wisely and partnering. And the New York Genome Center is taking money not only from the association, but from lots of other philanthropies as well, and putting it together in really effective ways. We talk with those other funders. I think we can formalize some of those discussions and formalize some of that measurement so we can be even more impactful with our investment. And there's also an issue about accountability. The Ice Bucket Challenge is a perfect example of this. Millions and millions of people invested in ALS. They donated to help people with ALS, and we owe it to them to explain what happened with those funds and what the benefits are. And when it comes time to talking about what our research agenda should be, in terms of broad questions, these are community questions. How much should we be spending on understanding what ALS is? How much should we be spending on finding a cure? How much should we be spending on helping people who have ALS now live with the disease in a more effective way? Those kinds of questions I think we can start to engage the community on in a broader way. We're also looking to not only spend our own money more effectively, but lure more investors, more private sector investors into ALS so we can fund clinical trials in a bigger and faster way. And then ultimately, of course, we want to improve the lives of people with ALS. We want to find a cure. So thank you. I'm going to stop here.
And today I want to talk about ALS in the age of genomics, and do this in the context really of a broad thank you, both to Hemali for the leadership she's provided in bringing the ALS genomics community together to create the types of scale that are necessary for the genomic interpretive work that I do. And then also to the ALS Association, and not just ALSA National, but also to the local New York chapter, who, using money from the Ice Bucket Challenge, has funded lots of the work that I'll begin to share some of the preliminary results from today. So I think I have subtitled this Reason for Hope, and the reason for that is that we're all very excited about therapeutic developments that have been occurring for ALS. We were just all at a conference and met with pharmaceutical companies, and by last count, there are in fact more than 50 drugs that are at some stage of development directly targeting ALS. This is up from 5 when I first started in the field, and that's obviously a dramatic increase. As you'll see here, many of these are what we term molecularly directed therapies. That means that they are designed to go after the root cause of motor neuron disease, either by attacking a gene that we know is involved in the disorder, or by supplementing a gene we know that is lacking in the disorder, or getting rid of genes by lopping them out or supplementing them in some fashion. And if you were paying attention to the news coming out of the American Academy of Neurology, everyone in the ALS field is extremely excited by the interim analysis of one of these genetically directed therapies, something called an antisense oligonucleotide, which goes into the neurons of people who have mutations in the SOD1 gene and blocks the production of more SOD1 protein. And there was a question as to whether this type of a therapy might be successful for ALS. And based on the interim analysis of a small group of patients, it's quite clear that this drug is working and that it's slowing down the disease, causing it to plateau in some forms of SOD1 associated ALS. So I think all of us in the field have high optimism for drugs that are being developed that tackle genetic etiologies. And that leads me to the main point of my talk, which is that we need to know the genes in order to target them with these types of therapies. Hence the research that Hemali and I, Lyle, and other groups funded by the ALS Association are working on. So to briefly recap for those of you who are not ALS geneticists, we now have a lot of genes that have been discovered for ALS. This is the timeline. Around the time that I was graduating from high school, the very first gene, SOD1, was discovered for ALS. And since then, we've seen slow and steady progress and then accelerated progress over the course of the last decade in discovering genes that either very strongly cause ALS or influence how it presents and progresses in patients. And once you get to 24 genes or 32, depending on whose review articles you read, you have to ask yourself the question, why continue to discover genes? And I think that's illustrated by this next slide, which is if you take all of these different genes and analyze their functions and ask where there are overlaps, we should get some sense of the important pathways within neurons and within ALS that we should be targeting our efforts on. And that's exactly what's happened. So as we've discovered more and more genes, we've been able to zero in on problems with protein recycling, the cell's ability to clear out old proteins and to recycle them. We've been able to uncover the fact that how cells process a molecule called RNA is especially central to how ALS develops. How the cell takes things that were manufactured here in the cell body and ships them all the way out to the nerve terminals near the muscles has alerted us to the importance of the cytoskeleton and how axons transport materials. And then we've learned about some other pathways that are important. And one of the discoveries that I'll talk about today has put us onto a new pathway that hasn't been heavily investigated for ALS. And that is how proteins get modified so they end up where they're supposed to go within the cell. So when we first started doing ALS, or when I first started doing ALS genetics, sequencing technologies were very expensive, and so you looked in people who had familial ALS. And so here, in the very first ALS cohort that I collected, which was comprised of almost 400 patients, most of whom had sporadic disease and a handful of whom had familial disease, we were able to identify a gene mutation that caused the disorder in about 67% of people with familial ALS. As sequencing costs got lowered and cheaper, we were then able to look at those same genes in sporadic disease and found out that about 9% of people also had mutations, leaving about 91% of people with sporadic disease for whom we don't have a specific cause yet. We've now been focusing in on this group in particular, the ones where we haven't found something, to ask whether we can identify new genes from those individuals. And this gets back to what Hemali was saying about the importance of the number of samples. So basically, if you have a large number of controls, people who do not have ALS, and a large number of cases, in this case, people who do have ALS, and you sequence their entire genome, you can then use a statistical calculation to ask, are there any regions of the genome which are more affected in the cases, the ALS patients, compared to the controls? So shown here are all of the bright green stars, which are differences in the genome that don't cause problems. And then the ones in red are the ones that look like they might. And if you add up the numbers that are seen in the controls and compare them to the number that are seen in cases, you can then use statistical methods to identify this region as being a cause of ALS. So we've now applied that to increasingly large cohorts, and back in 2015, 2016, we're the first group to identify NEK1 and TBK1, these two genes shown here in red, using this new type of association test. Other groups have gone on to discover TUBA-A4 and a couple of other genes by using at least this type of a methodology in part. And then using the next slide's methodology, we've gone on to be able to identify this new gene called LG-ALSL as a new candidate for causing ALS. So how have we done that? So while the samples are accumulating, while we're sequencing more and more patients, we're also developing new computer algorithms to identify the gene regions. So shown here are two of the new approaches that we've developed. One where we do that same type of statistical test I just showed you in the previous slide, but looking at different sections of the protein, or by asking, what do we know evolutionarily about the different regions of the protein that tell us where to look, and so thereby focusing our laser vision on different regions of the genome. And by doing that, we were able to identify that gene I told you about before, the LG-ALSL gene, which has put us onto a new type of protein modification that we didn't know was involved in ALS before, and may yield a new pathway for us to talk to our pharmaceutical industry partners about designing drugs to affect that pathway. But as Hemali alluded to, the ultimate advance in ALS gene discovery are numbers. Numbers of patients, numbers of samples, number of genomes for us to analyze. All of these big data statistics rely on us having the power to identify small differences between the two groups that are informative. And so the ALS Association, partnering with the drug company Biogen, have funded my project, which is entitled Genomic Translation for ALS Care, or GTAC. For those of you who aren't geneticists, GTAC happen to be the base pairs that make up our DNA. And we really believe that by zeroing in on the genomes of people, we'll be able to figure out subgroups of patients, genetically driven subgroups of patients that will help us design improved therapies. These are the centers. We have multiple centers distributed across the United States, as well as the entire region of Scotland. And all of these centers are taking any patient that comes to their clinic who has ALS and motor neuron disease, following them for 3 years, every 3 months, to find out how they do over time, collecting tons of information about their disease, as well as important exposure and lifestyle factors that may help us get at the genes versus environment question, and then sending us lots of fluids and cells for us to store in a library for downstream testing. So most important, of course, is the DNA for whole genome sequencing, which in our particular project we thought was important to get the information back to patients. And so after we do our sequencing, if we find a mutation that we believe is causative, we then check it in a laboratory that is CLIA certified and then return it to individuals. And by doing so, we've actually been able to identify more than 45 people who carry the C9orf72 gene and didn't suspect it. an additional 15 that carry mutations in the SOD1 gene. And these individuals are now, thanks to this study, potentially eligible to participate in that clinical trial I was telling you about that looks like it's having good effect, and a sister trial, which is using a similar molecule for the other gene, the C9orf72 gene. So this study is kind of giving back to the patient community by letting people know about their genetic status and helping them get connected with trials. So the study is the fastest enrolling and ALS study in the history of the United States. So over this short period of time, we've been able to enroll now over our 1,000th patient. But we're not done. We really want to reach a goal of 1,500, which is the amount of money that Biogen has committed to doing the ALS sequencing. We don't want to leave any of that drug company money on the table. We want to be able to sequence up to the full 1,500 and are on track to do that over the course of the next year. We've also worked very hard with similar projects in order to make sure that we'll be able to overlap our data. And so in particular, we're working very closely with Answer ALS and Project Create, as well as our European colleagues who have a similar project called Project Mine. And again, Hemali has been instrumental at bringing all these groups together in order to be able to share data and get more bang for the buck. So what's our ultimate goal as we do this? So we're facing a group of ALS patients, shown here in red when they come into our clinic. And what we'd really like to know is what are the biologically relevant subgroups within the ALS population. We want to be able to find all of the green ALS patients that are from one biological pathway, those that are red, yellow, blue, so that we can basically parse them out and find individuals who have the same underlying cause. We have some clues about what those underlying causes might be. It might be neuroinflammation, glutamate toxicity, the protein recycling that I was talking about earlier, maybe the cellular transport group, and thereby be able to develop drugs that target each of these groups, so that we would be treating the neuroinflammation patients with an anti-inflammatory, the cellular transport patients with something that speeds up transport down the axon, and thereby bring precision medicine to ALS. So how do we find these groups? Just briefly mention, we're doing computer analysis of clinical data to see if we can discern biologically relevant groups from what we see in the clinic. And then what you'll hear more about when Lyle speaks is biomarker discovery efforts. Can we study the brains, the samples, the electrophysiology, the transcriptomes of people who have ALS, and use computer-assisted technologies to find those subgroups that tell us about the underlying cause for the next step, the discovery of the therapies for them. I'll go skip these in the interest of time, but the hopeful message here is that with increasing numbers here in this study, 12,000 patients, computer algorithms do a very decent job of predicting the path that someone's ALS will take. And by studying these biomarkers, these studies here are finding markers in people's spinal fluid and in the blood that allow us to predict how they might be doing in the future. I'll just point out one thing that the GTAC study has been contributing. We've been looking at the genes that are turned on and off in patient blood from our study. And we have discovered a gene expression profile that allows us to figure out who has the C9orf72 gene without doing genetic subtesting. And this, I think, will be very interesting as we look at other genes to help us figure out the underlying genetic cause and biological pathways. So I'll just wrap up by saying that there are many reasons for hope in 2019 in ALS, many of them spurred by the ALS Ice Bucket Challenge and the ALS Association and their funding. So the first of these is that molecular therapies are nearing clinical use and showing good results in clinical trials. As Hemali said, researchers are working together as they never have before. And out of that, new genes are emerging every few months that we're leveraging through artificial intelligence and other computer algorithms to bring new discoveries to the space. And so with that, I'll hand it over to Lyle and look forward to the great questions that you'll have at the end.
So I'm going to talk to you about our post-mortem tissue core and how it fits into the rest of this ecosystem. And in particular, how we have designed this not only to provide resources to our researchers in both academia and industry, but also what we're doing to really, really facilitate open science and collaborative science. And everybody talks about collaborating and consortia, but it's really hard to do in a real-time way that makes any difference. And that's really one of the things that I think we're most proud of. And I've kind of got my, I guess, acknowledgments up front because it's an unbelievable privilege and an honor. I get to work with all these super smart people all over the country with really diverse expertise, and we really do work together. And so these are the sites in our post-mortem tissue core that are collecting autopsies. And then it's not just about the scientists. We have this always ever-growing ecosystem of different foundations and other organizations, and it's kind of like alphabet soup sometimes, but it's becoming more and more simple because we're all getting in the sandbox together, and we're leveraging everybody's investment for everyone. Matt and I are also clinicians, right? We see patients, and we see ALS patients, and we love our patients, and we're not doing this at the end of the day to get publications or promoted. We're doing this because we need to treat this damn disease, and we need to do a better job, right? And right now is a very exciting time, and Matt alluded to this, because we've got a lot of stuff going on, and there's some preliminary data that looks like we may be finally having the cusp of some real treatments for certain types of ALS. But the fact is, if you were in a clinical trial in the past 30 years, you would have rather been in the placebo group, because really nothing worked, and some things actually sped up disease. And that's not acceptable, and we have all these researchers all over the world doing phenomenal stuff in lab with animal models and cell cultures and IPS, and it's this piece that we've been screwing up. This is a known figure. I actually should probably update it, but this is showing different sorts of things that were coming to clinical trials and phase 1, 2, 3, 4, and the point is, there's a whole lot of stuff out here, and nothing really makes it into here. And right now, again, we have some stuff with some exciting preliminary data, but we've been bit by this before, things that looked good, and then we bring them to a big trial, and they fail. And it's not just that they fail. When they fail, we don't always know what went wrong, right? So when a big trial fails, and I'll just give the example of ceftriaxone, okay? So ceftriaxone was a drug a few years ago. There was a lot of excitement, some early phase trials that looked like it was going to do something. Big phase 3 trial, totally failed. And when it failed, we weren't really sure why. So did it fail because it was just a dumb idea, right? Was it just a wrong idea that we were just mistaken and somehow the animal data was wrong? Or did it fail? Maybe the drug didn't get to where it needed to go. Maybe it didn't cross the blood-brain barrier. Maybe it didn't have the effect. Matt was talking about different processes that we're trying to affect. This one was supposed to upregulate something called, EAT2, and maybe it wasn't doing that. We didn't know. Maybe it only works for some. Maybe that's the problem, right? Maybe it's because when we enroll patients, we enroll people with everything, and yet these treatments are coming from models in the lab where it's a particular gene or a particular abnormality. And so the other thing that we realized is Matt talked about genetics. Okay? Only 10% of ALS patients are familial. The other 90% are what we call sporadic. And are these animal models, if I have a mouse that has a mutation in a particular ALS gene and I do something and it gets better, is that actually relevant to patients who have sporadic disease? And how do we figure that out? It might be, it might not be. So what we've realized is that we really need patient-based tools that we can validate these things with. Post-mortem tissues, cells from our patients, biofluids from our patients, so that we can tell, is what we're seeing in the lab really relevant? And also, can we tell who it's relevant to? And Matt sort of talked about this a little too. And when we went looking, we realized, there really was no source of high-quality human post-mortem tissues for ALS research. There were these things for Alzheimer's, for Parkinson's, but guess what? They don't care about spinal cord. We care about spinal cord. There's certain regions of the brain that control your movements that we really care about. And there really was no resource. And so that's why we designed this post-mortem tissue core in the beginning. And our goal was just that, was to provide the resources needed, the tissues, the associated data for ALS researchers. We wanted to make sure that we were maximizing the use of every case. If a patient donates their brain to me, holy moly, that's like the ultimate donation, right? I better be sure that we're using it responsibly, that it's being used for the most possible things, that the most researchers around the world are benefiting from that data. I mean, otherwise, it's just not acceptable. And so we designed policies and principles and ways to do a dissection of a brain that's optimized for ALS research, so we get the most specimens for ALS research, so that we analyze it, so that we get the right data specifically for ALS research. And it turns out this was really hard. And You've heard the word harmonize a few times, right? And at the beginning, there were all these meetings, and Matt mentioned some of these other efforts, and all these people get together and say, oh, this is the most important clinical data, and this is what we have to do under the microscope. And one of the things we learned in this process was harmonizing doesn't actually mean doing things the same way. It means deciding what is your data set that needs to be consistent. What is your minimal clinical data that has to mean the same thing? What is your terminology? What, are you referring to the same things? But then it actually adds value in a federated model if different places are doing things slightly differently, if different places are doing more of one thing or another, as long as you have a basic set. And so at one point we just said, screw it, we can't talk about these details anymore. We have to bite the bullet and do it, and some stuff's going to work and some stuff isn't going to work. And what we figured out was the way to then adapt, right? So it's not about what the facts are. It's about looking at the research, looking at what your researchers need, and understanding how, as you go, to change your policies and what you've harmonized. So I'm not going to go through this unbelievably detailed thing, but just to show that it is crazy detailed. So we have a dissection that produces the maximum number of samples that are needed from the regions that our researchers need. And how do we know this? Because we surveyed them, and we said, what do you guys need? We have online databases that keep track of all of this across all of our sites, and the tissue is stored locally, and it's all barcoded. And we have standardized slide sets and standardized ways that we call everything. And most importantly, if you use tissues or resources or data from us, it's yours. So everything is provided, no strings attached, no authorship requirements, no ownership, no intellectual property. This is for academia, this is for industry. Because this is all about getting the tools into the researchers' hands. It's not about necessarily getting the credit or being the one who gets your name on the paper. We have a detailed set of neuropathological data, slides that get scanned in. This is the kind of thing that we standardized across our sites. We have unbelievable genetics done here by the New York Genome Center and funded by the ALS Association, the Tau Foundation. So every single case gets whole genome. Every single case gets RNA analysis on multiple CNS regions, and all this data is made immediately available as soon as it passes quality control. There's no embargo. When new data is available, it is in the hands of researchers. No ownership, no authorship, industry, academia, everybody. The NYGC has made this unbelievable platform, this metronome thing. You can sit at your desk during lunch, and you can look at this data, and you can analyze it yourself. And once a week, we have phone calls where Hemali's team is so sick of me, but it's such an unbelievable thing because these phone calls are to keep making metronome better, and the way we do that is because we get requests every week for data, and there's all these evolving research programs, and we can say, hey, this is what the researchers need, and we continue to add functionality, and we continue to improve the program. You can go on the website, you can play with it right now tonight after you drink some beer. It's really, really fun. All the data is available in raw data files, and again, freely and openly available. One thing we hadn't anticipated, which we're kind of getting screwed, and we got to fix this, is that there's a monetary cost involved in making data freely and broadly available because of all the ALS consortium data sets. Ours are the only ones that can't be archived, right? Because you can't archive them if you're going to have them be available anytime, and it's a lot of data transfers. All of us work together, so then we send samples to them. They send them to this guy, Matt's lab, and he does some of the analysis for us, and he's also one of our sites, so everybody's really working together in every aspect. So that's a lot of stuff, right? And it's insane, and this is just what's involved in getting data into the system. This isn't about getting tissues into people's hands or any resources into people's hands, and it's all done at different times, and it's about having highly curated research information, but on some level, this is more like a supply chain problem than a science problem, right? So this is like what's involved in one single autopsy and all these different stuff that has to happen. And the interesting thing is, this is not the hard part. And I think what we do different than all these other studies and all these other collection efforts is that we have figured out how to operationalize the other side. And the other side is getting the stuff to the researchers and doing it quickly and doing it responsively and openly and making sure that the experiments are properly designed so they work. And so early on, we said, let's have a review committee and people will write proposals and we'll review them. And the fact is, I just told you, like every trial has failed, right? So I'm an ALS expert. Like how can I say that your idea is better than your idea when everything is failing? What I can say is what's responsible research, okay? Are you requesting the right numbers of samples? Can you do the experiments you say you're going to do? Can you show me that your stain works, that your assay works? If not, we have test sample sets that we send you, and I help you debug your assay, validate your antibody, whatever it might be, to make sure that your experiments work before we're giving you scarce rare samples. We abolished our review committee because that was slowing things down, and we said, you know what, we're not going to review based on science, we're going to review based on principles of experimental feasibility and appropriateness, as I was just describing. And guess what? We're going to do this rolling. And rolling means whenever they come in, we schedule a phone call and we talk about this stuff and we start your project. And we might give you samples and data many times as your project evolves. And rolling right now means me, and I'll just tell you that on the train ride from Baltimore to here this afternoon, I got 3 new project proposals. And they're super cool, but this is unbelievable how quickly, you make stuff available, people use it. We say yes to everybody. We don't say no, but we say yes and we help you use things responsibly. And this is for academia and industry. And so if there's a postdoc in Indiana who has a new idea and rather than write a grant and get like 9 samples and learn how to pipette and do something on some small scale, we can probably get him the data and the resources he needs to get an answer within the next couple of weeks and then write a much bigger grant and really move things along faster. So how are we doing? The fact is, we have given out thousands, I stopped counting, it's many thousands of tissue samples and slides. On last count when we summarized it, which was a month ago, 650 terabytes of the raw genetic data have been transferred from this effort. This is supporting more than 100 collaborative ALS projects around the world. I write letters every week. I wrote one for Dr. Phatnani yesterday, supporting grants, follow-on funding for these programs. This is just, so we're trying to figure out, Neil's really interested in metrics and so are we, like how to automate, the impact this is having and how ways that we can sort of track this without having to do a lot of work. Well, there's this program, I just took 10 of the manuscripts, there's dozens and dozens that have been published, but 10 manuscripts using our core, and just those 10 have now been cited over the past couple of years, almost 1,000 times. And so this really is working. These are just some examples of the stuff people's using it for. This is a nutty list, and a lot of the stuff on the list is stuff you guys have never heard about because, again, we're not reviewing for science. I don't know what's going to be a good idea, okay? This is not even a complete list. I just couldn't justify a second slide or a 4th column. But a lot of this stuff is not stuff that's necessarily mainstream. Similarly, this is a list, and again, not updated, and I just got tired of some of the labs, industry and academia around the world that are making use of these resources. And again, this is the people, so this is tissue and slides. This is raw genetic data from the New York Genome Center. And my research coordinators got mad at me and they're like, we're not putting any more pins in maps. So like a big pin means there's like 7 of these things in Israel or I don't know what. And again, when they ran out of room, they said, we're not doing that anymore. So, but it's remarkable. If you make data available, if you make resources available, guess what? People use them and they use them and they figure stuff out. People use Metronome specifically for what Matt was getting at, to figure out, like if you're doing an experiment and you have a particular pathway and a particular target, you'd like it if I can send you samples from the right decedents, right? The right patients who had a whole lot of this problem or none of this problem or different levels of it and different levels of their brain. And so we can do that because all the genetics are linked back to all the tissues. I want to give you an example of the way these projects evolve in sort of a true story way. That I hope will drive home how much fun this is and how amazing and what a great privilege it is to be part of this. So this was one of the first bigger studies that used our tissue core when we started in 2013. This is this human endogenous retroviruses. This is this idea that millions of years ago, your ancestors were infected with these retroviruses, which are like HIV. They're similar to that. And you're not infected, but those things left stuff in your DNA, this junk DNA. And in some cases, in some diseases, it gets turned on. And when it gets turned on, it can contribute to disease. And when we started doing this, this is like the X-Files, right? This is not a acceptable disease mechanism. But lo and behold, using our tissues, Dr. Nath and his colleagues showed that ALS patients, a significant portion of them have more of this stuff and have it turned on. And he took a protein from this endogenous retrovirus, he put it in a mouse, and the mouse develops what looks like ALS. So when this paper got published, like 3 really large pharma companies called me because they were pharma companies that had drugs for treating HIV, right? Because these are not infections, but they are retroviruses. And they said, hey, we want all your tissue, and we're going to do our own RNA-seq analysis for retroviruses because they thought that their existing drugs were going to be things that could be treated. And we realized, oh, crap, that's going to like deplete everything we have, and you guys are all doing the same thing, and that doesn't make sense, and that's not responsible. And so Hemali, and by the way, to detect these things, you have to do it, the RNA, in a more expensive and harder way than normal. And the ALS Association had funded Hemali to do the normal way. And so we just sort of called them up and we said, hey, this is the story. Can we use your money to do this expensive thing and really make it broadly available to everyone and And that way we don't have to waste all these tissues and we can make sure it's highly curated. And ALS Association said, of course, that's a great idea. And so that's what we did. And as this evolved, there was a conference in 2017 on endogenous retroviruses and disease. They do things in other parts of your body. They devoted a whole session on this to ALS, and Dr. Nath presented his work. And at this meeting, and they made me chair the session even though I don't know anything about it. And at this meeting, we met this person, and this is Molly Hemali at Cold Spring Harbor. And Molly is a super data wonky person who analyzes this stuff, and specifically these retroviruses. And she said, hey, I'd love to look at your ALS data. And we said, tell you what, you can have all of it, all the data. And she kind of looked at me like, what? And I said, yeah, you can have the entire data set. And she said, well, if you give me the data set, I'll give you back my analysis. And everyone else can use that who's interested in this area. And she also warned me, she said, by the way, is it okay if I find things that aren't retroviruses? Because when we analyze this stuff, we tend to find other things. And she thought that would be like a bad thing. Like, no, you can. I'm like, of course, that's great. As this evolved, we had a lab meeting, I'm calling it here, where we basically brought in labs from all over the country who are working in this area together. This world is our lab, and we talked together about how we could help each other analyze data, fix problems. Some of them were having different kinds of issues. It was the kind of thing like when you're a kid and you imagine what science is supposed to be, like this is what it's supposed to be, okay? I mean, people from all over getting together, leveraging their expertise. So Molly analyzed our data, and in line with sort of what Matt was talking about, what she found was that if she lets her fancy-schmancy math look at this RNA data, that ALS patients fall into clusters. And you can define different clusters based on different things that are wrong with them. Like this cluster might have retroviruses. This cluster might have problems with one kind of cell type. And that's interesting, right? Because maybe our trials don't work because we're enrolling everybody. And maybe what we need to do is if your drug targets this cluster, well, that's the ones that you really want in your trial. And that's the ones that you'd like to cohort select for your trial and know that your treatment is working for them. And so what if we could find a way in biofluids or muscles or something, because I can't do like brain biopsy and comes to see me in clinic. I mean, I could, but it's not ethical. If we could find some sort of a peripheral correlate of this. And so what researchers really wanted was biofluids from the same people that we eventually get autopsies from. And so guess what? We had already known that because that had been something we'd been being asked for a while. And we had gone again to the ALS Association and said again, hey, this is really what we need to move the field forward. And they said, you know what, that's a great idea. Why don't we fund biofluid collection at all your sites? Again, freely, openly, broadly. We are now in the process of expanding this to longitudinal collection, so we can take samples from people over time and see whether these things change. And Matt mentioned, this is like alphabet soup, I know. So CREATE is yet another one of these efforts that collects genetics and data and I mean, there's a whole bunch of these, okay? Now, it's gonna take a while till we get enough samples to do anything with this because, our patients hopefully are living longer and longer and not, anyway. But this is a very mature effort with a whole lot of longitudinal fluids, very well characterized and extremely highly curated. Like many similar efforts, they weren't really operationalized to give this stuff out. We said to CREATE, tell you what, if we give you a coordinator to help disperse these samples, will you give us access to these samples? And they said, yeah, that's a great idea. And so we did that. And so our researchers who had things they were looking for in samples that were from the CREATE, and again, partially funded by the ALS Association, so this is leveraging these different bins of funding. And so what is this slide? So a couple months ago, I was at their meeting because of that, because we have these collaborative projects that were involved. And all of a sudden, there's a presentation from somebody at the CREATE meeting using our target ALS data from NYGC. And why? Because we make it freely available. And they took our whole data set, and crazy enough, the story that he's telling is a retroelement story that they happen to find in the data set. But even crazier, they have this great bunch of tools, these genetic analysis tools called the St. Jude Cloud, designed for cancer. They're now building an ALS platform of analysis tools, and they're using our data to help do that. And our researchers are going to have access to our data linked to our tissue samples, linked to the genetics here, and using these analysis tools freely and broadly and openly. And I'll tell you, I don't think 5-10 years ago, things were very siloed, right? This is literally everybody getting together and using all the same resources, and it's fun. Back to Molly. So Molly, based on this analysis of our data, this idea that she could find different clusters, which got a preprint published, she won an award from the Chan-Zuckerberg Association to continue this research. They just had their big kickoff meeting. They invited me out there because, I hate talking. I'm sure you realize that. But no, to basically talk about how we do this open sharing and this, this kind of open, broad science and see if we can do it on a bigger scale. And by the way, so I just want, these are just a couple examples of some of the raw data requests we get, and these ones are specifically from industry, right? And so these industry people want our data to figure out how the different groups are different. This one, look at this. We would like to reproduce the results published. So that's this preprint that hasn't been published yet. I got this thing while I was sitting at this meeting in Florida, and the company that sent this one was at that meeting, and I walked over and I was like, what? And they said, yeah, everybody's got that preprint. The whole meeting's talking about it. And it's just invigorating. And everyone in ALS is using human tissue more and more, and our data and our tissues, and it is really becoming one big sandbox. Even beyond ALS. So there's a big association, as I'm sure most people in this room know, there's a spectrum between frontotemporal dementia and ALS. Other disease foundations looked at what we're doing and they said, yeah, this is what we all have to do. And we've been working with several of them to figure out how to expand what we're doing in ALS into other diseases, particularly frontotemporal dementia. One of the foundations we've been working very closely with, and in the next few months we will be rolling out an expansion of what we're doing into FTD and other tauopathies. And we sent surveys out to all the researchers around the world, like hundreds of them, and asked them the same sort of thing. What do you need? What's the data? How can we make this work for you? We asked them about what kinds of patients do they want. Interestingly, there's a lot of overlap in the genetic kinds of patients and in the controls with what we're already doing. We asked them, what are you going to do? What kind of analysis? And there's not only a lot of overlap, but there's some opportunities for centralizing, beyond the genetics, centralizing some expensive other analyses. High throughput proteomics, okay? Things like this that both of our communities need, that if we could do it rigorously and centrally at one sort of CRO-like facility and make it freely and openly available to everyone, it conserves resources, it saves money, it speeds up everything, makes the data available. And so they're very excited about those possibilities. This is going on right now. This is this year. This is our plans to hopefully roll this out. And I'll just tell you that I can't tell you much about it, but there are even plans beyond this to try and operationalize what we're doing to other diseases, because what we're doing is more principles, more supply chain, more figuring out how to share and share openly and leverage different small investments for everybody. And so, what's the pie in the sky, right? It's kind of corny to say, but the world is our lab. And every day I get these requests, and somebody calls me up, and yeah, we've got this great idea, and I'd really like to get 8 brain samples from you to test it. And I say, sure. I can give you 8 brain samples, but I'll tell you what, there's somebody else already using our data and our samples, and I can give you the data from 1,000 samples, and if you're willing to talk to them, I won't talk to them unless you tell me to. I'm sure they'd collaborate with you, and you can have all that data tomorrow, and nobody ever says no, okay? And so the most fun of this isn't actually the scientific results, it's these collaborations that form in real time every week as the research is evolving. Academia to academia, academia to industry, involving different foundations funding. And actually, I said nobody ever says no. One person said no. But beyond that, nobody has ever said no. And I'll tell you, so I mentioned Hemali. So this people who I said at this meeting, like, what are you doing to me, like when they sent me this thing here? Okay, so now she is now directly collaborating with this pharma company. Another pharma company who has another set of drugs that had relevance to this is now collaborating with Dr. Nath, who started the HERV thing. And in fact, using samples from the CREATE consortium, which isn't even part of our efforts. And this is the way that this is evolving. And I'm very grateful to be in this position and to be able to do this kind of thing. And it's because everyone is working together and working openly. And so with that, thank you. And I will say one thing, which is that although I do thank all of the organizations and scientists involved, this is really what it's all about, because we are depending on brain and spinal cord tissues from our patients when they die. So I take care of patients. I meet their families. I talk to them. Matt does as well. We enroll them in clinical trials. We really, when our patients pass away, it's very hard. And then when they decide that they would like to donate to this research, that is the ultimate donation. There is no price tag. And it's a privilege and an honor. And we couldn't do any of what we're doing without our amazing patients. So thank you, guys.
So in all other complex diseases, it's kind of widely known that majority of risk variants are situated in regulatory regions rather than in genes. And somehow I never heard this kind of talk from ALS people and never seen it in the papers. And I wonder why.
So whole genome sequencing on the scale that's needed to identify regulatory regions as opposed to the parts of genes that make protein is just now something that we're able to do. And so the other complicating factor is in order to infer that a regulatory mutation is having an effect, you need to be able to see, in most cases, a change in protein expression. And so you have to have a pairing between the genome sequencing and the genes that are turned on and the genes that are turned off. So we're doing that in living patients between their genome and the gene patterns in the blood. But then the post-mortem tissue core and all the sequencing that's been done here at the New York Genome Center allows that to actually happen in the brain. So those investigations are coming. We just haven't had the numbers and the data to do it until very recently.
My wife has ALS, and she's under the care of the neurology department at HSS. Should I assume that, like her, just having genome sequencing and analysis performed, or, you're looking for data? So I just was wondering if it's routine. Yes, every ALS patient at HSS, let's say, is having their genome sequenced and analyzed.
So most clinics don't have whole genome sequencing available for ALS patients because of the cost and expense. So many ALS clinics will check the two most common genes, the C9orf72 gene and the SOD1 gene, maybe a handful of others. But not every clinic has the protocols and the research staff to basically collect the DNA to do whole genome sequencing. So I don't know for sure whether Dr. Lang and his team are doing that by some other mechanism. But if not, and anyone wants to participate, we could put my email address for the GTAC study, which is available to anyone in the New York area or anyone who's close to any of the other 13 sites to participate.
So HSS is also a partner of the New York Genome Center sequencing efforts. So depending on when she donated her samples, we would have done the whole genome sequencing on it. But the bottleneck here is the interpretation and the returning of the results. And that is something that we hope to partner more closely with Matt on.
And actually, it's not trivial also that, so a lot of research being done on ALS genetics is just that, it's research. And a lot of times the genetic results are not being done in what's called a CLIA certified space, right? And so there's rules about what you can and can't return to patients if it's a research result or a CLIA certified result. And this is very frustrating to us. And I'll tell you, for example, when the C9orf gene, which is the most common gene involved in ALS, was first discovered, and we had all these research tests for it, nothing that was CLIA certified, we tested all the autopsies in our core at Hopkins. And as would be expected, a significant portion of them had this gene, including about 10% of the sporadic cases, which is what literature would show. And Hopkins would not let me tell their families that they had a genetic disease, and I understand that. When we talk to patients and families with ALS, and we talk about the regulatory hurdles, which, and PHI, like private health information, almost unanimously, the families tell us, we don't care, use what you need, share what you need, we want you to make progress. One of the things that we're working very hard on is to figure out ethical and responsible ways to, when appropriate, return research results, genetic research results to families. And it's a very difficult thing, and it's a moving target. And when we have researchers in other countries, it's especially difficult because the regulations are different everywhere. So,
my friend has ALS, and he recently started stem cell therapy. I mentioned that stem cell therapy wasn't mentioned here. What is your opinion on that?
Sure. So there's a lot. Stem cell therapy means a lot of different things, right? So one of the things that we've been doing in ALS research for a while is that we can take skin cells from our patients in clinic or anyone in clinic, and we can grow skin cells called fibroblasts in a laboratory, and we can actually turn those skin cells into what are called stem cells, meaning cells that can then become other things. And then we can take those stem cells and we can turn them into brain cells, neurons, astrocytes, the kinds of cells that are in the brain. And this is something that is often more and more being used as a research tool, meaning if I have these stem cell derived brain cells from lots of different ALS patients and different flavors of ALS, right? Genetic and fast and slow, and this person's, different kinds of ALS. I can look in a dish and see what's different, and I can see maybe that they've got something wrong in those cells, and I can put drugs into those dishes, and I can sort of do a clinical trial in a dish. And this is one of the tools that's being used more and more that we're hopeful is going to translate to effective clinical trials. Now, that's different than what you're talking about, which is stem cells for treating patients. And there are some ongoing trials. They're small trials looking at that. And to be totally honest, and I'm happy to hear Matt's opinion too, I don't think we quite understand the rationale. And there is some data that's sort of interesting and possibly exciting in a couple of instances, but I think that the jury's still out. And I'll tell you, I don't know about you, but I have lots of patients who have gone overseas because some clinic in somewhere else has said, oh, you give us $50,000 and we'll give you some stem cells. And that is not something that is we recommend ever, because it's never useful and they're never followed and they're not tracking the data, and they come back to us and they've actually often been harmed. And certainly their pocketbook has been harmed. So whether or not these trials, we are very hopeful that any trial in ALS is going to work. I don't think we know yet whether the stem cell therapies are going to be successful.
I'll say that stem cells will be a part of how we treat ALS, I believe. I'm optimistic for that. The stem cells that are being tried right now, I worry don't live long enough and don't manufacture enough growth factors, which is how we think they're going to work initially. And those are the ones that have been the best studied and are in clinical trials. The ones that are being given to patients in clinics around the world, and some including here in New York City, I don't think are effective. I haven't seen any.
But what Matt's talking about too is using these stem cells basically as a vehicle to deliver some mechanism, right? If we think that patients are missing something, we can perhaps engineer a stem cell to make a lot of that thing and put that cell back in. And yeah, we're optimistic about everything. We don't know what's going to work, but I don't know the answer right now.
Thank you.
Mm-hmm.
Hi. So you mentioned that a lot of the clinical trials in the past have not been successful, but then there's been this great hope with regards to the new SOD1 clinical trial that's going on currently. So I was wondering if you had visibility into whether or not this clinical trial in particular has kind of changed the way that they are utilizing all of the data that you've been talking about throughout tonight, and if you have more optimism with regards to this particular clinical trial.
So I don't think Lyle and I are either in a position to comment with particular insights, because we are part of it, and one of the things that is very important is keeping all of the how it's happening and the patients and stuff blinded to the effects and that sort of thing. What I'll say is my reason for optimism, not just for that trial, but for others, is because we're targeting what we now have a much deeper understanding of the underlying mechanisms. And so these therapies are targeting the mechanism. It used to be that we thought we'd just be able to find a magic drug that helped neurons stay healthy and happy no matter what was attacking them, right? And I think many of those early trials that failed were kind of general neuroprotective strategies. And now we've kind of zeroed in on some key pathways. And the other advance I would say that the SOD1 trial is benefiting from is something that we call a target engagement biomarker, meaning that we know that the antisense oligonucleotide for SOD1 is doing its job because we've gone back and measured the amount of the protein in the spinal fluid and have seen it go down, 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 down as you give the drug. Many of the trials that were done before, including the ceftriaxone one that Lyle used as an example, we don't even know if it had the desired effect of raising or lowering the molecule we were after. So I think we're smarter about how we go about the interim step, and we're smarter about the pathways that are being targeted. And I think that's what's driving at least my optimism.
But to specifically answer your question about whether these sorts of resources are helping these trials, so this antisense in SOD1 in particular, this is not the first trial. There were a couple earlier, phase 1 safeties, smaller doses, just figuring out. So the FDA was worried, you have to make sure you're not going to hurt somebody, right? And how do you do these kinds of trials? And so the very first antisense SOD1 trial, which I'm now forgetting what year it was, but But I was the site PI at Johns Hopkins, and so I was, overseeing the patients. And this is one that they were getting a tiny little dose of medicine, and we didn't expect it to do anything. It was supposed to be safety, okay? And it was the very first sort of gene therapy trial in a neurologic disease that the FDA had approved that I know of in human disease. And so a couple of the patients who were in that trial subsequently passed away. And we did their autopsies as part of this work. And then we took their tissues and we were unblinded after the trial, so we know who got drug and when they got drug, right? And so then we coordinated with the main PI of the study and the company that was doing that study to test those tissues because we could detect the drug in the tissues and we knew when they got dosed and when they died. And we also could measure SOD1 in those tissues in different, so they were getting it through a catheter put in the lower part of their back. Well, we could take tissue from the lower spinal cord and the middle spinal cord and the upper spinal cord and the brain, and we could see if we could see drug in different regions. And these are the sorts of things that have been done in animals, not in people. And that data, even though it was only the 2 patients, was helpful in sort of thinking about for the next trial, what's the proper dose? What's the half-life of this drug in human tissue? So it helped to inform those later decisions in the other trials.
Question here.
Yeah, what is the basic definition of ALS? Because the way you've described it, or with the studies, it sounds like it may be 20 or 50 diseases that are...
Right. So ALS is a clinical diagnosis, okay? ALS is something that there's not a blood test for. That's one of the most difficult things about it. Being an ALS clinician, if you come to see me and I tell you you have ALS, I'm telling you that you have a fatal disease and we don't have a cure for it. And that diagnosis is based, unless you have one of these genetic forms, on clinical exam findings. And so there's a certain set of, it's painless progressive weakness, right? It's a disease of progressive weakness. When it involves muscles of breathing and swallowing, that's when it becomes fatal. But there's not anything except for the couple genetic forms that is a lab test. And that's why when we're doing these studies on human samples, it's also important to know that we have apples and oranges, right? And so when we have these autopsies, they all get a detailed neuropathological evaluation. And guess what? Even the world's experts in ALS and in FTD aren't always correct. And you don't want, if you're doing a study and you're using 10 samples, you want to know that they're all what you think that they are. And so part of our rigorous analysis is making sure that what we see in the brain tissues, right, because the confirmatory diagnosis is that, is what it's supposed to be. So there's different pathological changes. There's loss of motor neurons. There's a certain cell called, astrocytes that become reactive. There's different special inclusions, little basically stuff in the cytoplasm of different kinds of cells. So there's pathological diagnostic criteria for ALS, but during life, it is a clinical diagnosis. It is based on, and again, Matt was talking about, biomarkers for different trials. We would like to have, so it's a very protein disease. We see patients in our clinic who come in and they've got a little bit of weakness, okay? And they have ALS and, they may pass away 2 or 3 months later. We see other patients who come into our clinic with that same little bit of weakness and they come back a decade later and they still don't look any different than when we met them. And both of those people have ALS. And so because it's such a protein disease, there's a lot of noise in these trials. And so biomarkers aren't just about what's the mechanism, they're also about, can we predict who's fast, who's slow? Can we make sure that when we have a placebo group and a treatment group, that we're enrolling, matching in the right kind of patients? And the ALS Association is also funding these efforts right now for what are called platform trials, new ways to design trials, to adapt trial designs, to use more than one drug in a given trial, to figure out how to do trials better with less patients and reduce that noise.
To get back, though, to your point, which is that ALS may turn out to be 20, 50, who knows how many different diseases, we are fortunate in knowing from the pathology that they all seem to converge, no matter what the inciting feature is, they all seem to converge on some similar pathways. And so when we look under the microscope, we see clumps of a protein called TDP43. And so if we can kind of unlock how each of those different mechanisms leads to the problem of TDP43 and design drugs, then we have a shot, at least, of having a molecularly directed therapy that actually treats multiple different causes, even if in some cases it's inflammation, protein recycling, some of the different pathways that I showed. So they all kind of funnel down into one pathway that if we can turn back that clock it kind of may not matter as much that there are all these different forms.
But you want to know what a mess it is? These same pathways are abnormal in types of dementia and in other unrelated diseases. And we still don't understand why it is. So patients with a C9 mutation, one family member might get ALS, one might get dementia, one might get both, one might get ALS and Parkinson's disease. And these pathological findings and these general cell mechanisms that these funnel down into, we still, what we call selective vulnerability, meaning what is it about this patient that made them get ALS and this one get dementia, is something that we don't understand.
I think we'll give the speakers another round of applause and thank them.